Thank you all for joining us today. The following webinar is brought to you by nonprofit organization iExplore. I am honored to be introducing our seminar presenter today, Ryan Kern, who will be continuing his biology series. We already have 3 seminars, so if you missed those, make sure to check those out on our YouTube channel. So this will be our 4th installment in Ryan's biology series, and without further ado, I would like to hand it off to Ryan.
Thank you, Kathy. I'm so excited to get started today, and I hope all you guys are really excited too. So, like Kathy said, my name's Ryan. I go to Troy High School. I'm going to 10th grade this fall. And over here is a water polo ball because I really like to play water polo. And another hobby that I have is I like playing the piano. I like performing as well. And I really enjoy that. Another thing I enjoy too is biology, which is why I'm teaching this seminar. And I'm really interested in it. And in the future, I want to eventually become a doctor. So the topic of today's seminar is gene expression, how genes become reality. So we talked a little bit before, we have a lot of background information going into this seminar, right? We talked about DNA, we talked about evolution, how it's tied to DNA. We talked about like a lot of sometimes the traits in the DNA that we get from our parents, stuff like that. So we have a good amount of like background knowledge going into this seminar. And even if you don't, I'm still going to walk you through it too. But this is kind of bring together some of it. So if you definitely did not didn't see my last seminars, make sure to check out our YouTube channel as well for them. And it's bringing it together to see how DNA will go to protein. We have the information, but how do we actually make it into something that is physical, something that we can see, that we can feel, that we can touch? How does it go from being a piece of information into that? And that's what we're going to be talking about today. Here, like I said, the DNA, remember, stores a lot of our genes, which has these traits over here. These are all examples I use in past seminars. So we have the different types of hair, very, very coiled versus just straight hair. We have this thing called the widow's peak here, which is a trait seen in some people and it's not seen in others, and is passed down from parents to offspring. And another thing is we see height as well. So all of these are all controlled by our DNA. And this makes sense because what we're going to talk about in this lecture is how that DNA will make the proteins that will make this widow's peak, that will make you taller, that will make you have these different kinds of hair. So it really is really connected, and it makes sense once, I hope after this lecture, that you listen to this and you really start to see the connections and understand a lot better. Our first question of today is, why does a protein look like that and not like the thing below? Remember, we talked about ribosomes before, and they always look like this, and I always used this before. And so why does it look like that? So A, it's not actually a protein. B, it's just a more detailed version of a protein. Or C, it's a random photo that I got from a rainbow. We'll give people a couple more seconds. Yeah, I think a good number of people have voted, and so we can stop polling. Yep. As always, you guys never fail to amaze me, always picking the right answer here. It is just a more detailed version. So we look here, correct, right? And let me actually go back to this picture here, right? So we see this, and I'm going to explain each of these parts here. So you see this little spiral. I'm going to explain what that is, but all you need to know is that it is a protein. But what we do is when we use them for like diagrams, since it's so complex, just for the sake of the diagram, we make it like one glob, then one glob, and we just put it together to make a protein, or to make a ribosome, excuse me. And so these are like proteins that make it up. And just for understanding's sake, it's a lot easier to really just visualize it rather than having this super complex structure and then you put it and try to have a diagram. Because the main point of this diagram is to help people understand just the main idea behind it and what's happening. Here on the right, you may be wondering what this is, and this is a little hint of what we're going to be having today. And yeah, so just keep it in the back of your head. We'll bring it back eventually. So the first thing we start off in every protein is this amino acid sequence. Remember, a sequence is just like one after the other, all connected. So we talked about how, and we're going to talk about more about how the codons, remember, 3 nucleotides in one DNA. So you have a stretch of DNA, then you have, it's cut up into segments of 3, 3, 3, 3. One codon is like a word, right? And then each nucleotide is like the letters in the alphabet. So each codon will code for one amino acid, and that's just how it works. It'll always go to that codon, and then that's just how it works. So this amino acid, all you need to know is just all connected together. It's like one long string. Then it starts to kind of fold and coil a little bit. So remember how we talked about supercoiling? It's sort of like that, but it folds in different ways because DNA just mainly just keeps winding and winding and winding. But this one is going to like fold and coil and twist a lot more, so it can form more of these complex structures rather than the chromosome, which is it's complex, but it's also simple in the way that it's created is very just like you keep turning the DNA. So, and that's another reason why proteins are so important, is because since they can become so complex, that's why they're so good for so many different kinds of tasks, right? We said how proteins are not just like one set thing that they can do. They can do many different things, like robots coming from a factory, right? They can do a huge amount of work, a huge variety of work. That's the same thing here. And so here we see there's 2 types that it can fold into. It's the pleated sheet. So if you've ever seen, like, I don't know if many of you have seen this, but there's these like Post-it notes, and if you pull them out, it looks like accordion. Oh yeah, that's a better example. So you have an accordion, you pull it out, and you have like all the kind of the diagonal, like the diagonal plates like this, and then they can kind of come together. So that's sort of like what it looks like. So it has these like these squares that are all like this, right? And they're all connected. That's called a pleated sheet. Then over here we have a helix, and this kind of looks a little bit like DNA, but it's just a helix, and it's called alpha helix. Notice that in total there's about 4 different levels of protein structure. So what does that mean? That means that this is the most basic, the most simple structure. It's just the amino acids. Then we have, it can be either pleated sheet or alpha helix. That's the second most basic. The third is called tertiary. So if we look at the root word, we see that tertiary means like 3. So that means it's like the 3rd level. This is where it begins to fold. So you read here, it says 3-dimensional folding due to side chain interactions. Well, what does that mean? That just means, like I was saying, how the amino acids are going to start folding and start like wrapping and just like turning in all sorts of ways, because the amino acids are going to have like some attraction to each other based on their polarity, which just means, like, let's say, you know how like opposites attract? So there's a positive and a negative. They want to go together, just like that. Let's say one amino acid is positive, one's negative, so they want to meet together. So that's what makes these structures, as they get more and more complex, is just more of them being put together. So this is a 3rd level, remember? And the 4th level is, you could think of it as just 2 of these 3rd levels put together, and then they start to interact, and again, it becomes more complex. Now I want to go back to here, and I want you guys to take one more look here, and now we can kind of properly look at it with a kind of learned eye here. We see all of these coils here, those are the alpha helix, and this one, it's kind of hard to see, but in the other ones you'll be able to see better. There are some pleated sheets as well. So let me go forward a little bit. Yeah, this is just saying the same thing, amino acids. And you may see this notation, this little B here. So pleated sheet and beta pleated sheet is the same thing. But they use beta because it's kind of sort of like the opposite with alpha. And then this A here means alpha helix. And so you can see that they will actually, it's all one connected strand, but you can have the pleated sheets and the helix in the same strand, which makes it very complex because it can have all these different turns and different like twists and stuff like that that make it, again, once so more complex, which is why it can do so much work. Here is a picture of what a ribosome would look like if we didn't have the simplified model. So you can imagine if you were trying to like learn for the first time what a ribosome is, like you'd be like, what is this? Like, what am I looking at? But once again, we see alpha helix here, right? And then it's very hard to see since it's kind of zoomed out, but there's also going to be those beta pleated sheets as well. Now we're just only gonna answer like one question because today's seminar is kind of long, and then if you guys have any other questions, we can save it for the end. But yeah, we can answer one question right now.
All right, so our first question is, what is the difference between DNA and genes?
Good question. So DNA, remember, it's the bunch of nucleotides in a super, super long like string. Genes are just like segments. So it's not set. It's not like remember the codon is 3 nucleotides. It's not like that. A gene could be like 40 or it could be like 4,000. It really depends. And we'll see as we get like more in depth into this topic that they can really vary, and also how they vary will kind of affect like the end result as well, because it'll have like different amounts of genetic information. And usually there's also going to be like these start and stop portions, because remember, DNA is just one long strand. It's like super long. So you need to kind of cut it up, not physically, but with let's say like a stop sign and a start signal and a stop sign as well at the end to show this is one gene. Because if you didn't have that and you're trying to make proteins and you're trying to read it, how are you going to know? Like, what if you just you finished your first gene, but you keep going and you accidentally read another one? So it can become very complex, which is why we have these like start and stop ones. Very, very good question. I'm glad you guys were paying attention to that. Okay, but the main question is like, but how, right? Because we're talking about, oh, it can go to protein and like from DNA, but really, how does that happen? So I'm going to ask you guys too. How does DNA go to a protein? A, it's through transcription and translation. I think we can launch the poll now. B, a magical wizard casts a spell. That's my favorite one. C, it unravels and machinery directly attaches to begin building proteins. Or D, it recoils and supercoils to form a protein. This one looks a little more divided. Glad to see I can finally challenge my audience a little bit. Okay, we'll give everyone a couple more seconds, so make sure you guys vote. Okay, I think we can close the poll now. Okay. So this one was very split, and I'm going to reveal the answer right now. It was actually transcription and translation. So we'll see that C, the answer C, it unravels and machinery directly attaches to begin building proteins. That's very close, but there's one major part that we're going to talk about, which is actually showing that C is actually wrong. And D, no, the DNA does not like supercoil to form a protein magically. And I really wish it was a magical wizard. Okay, moving on here. Oh, there's nothing, but we're going to magically use our wizard skills here. And ta-da, yep, transcription, translation. So in transcription to translation, we're starting with the DNA, going to RNA, which I talked about. Remember, I compared them side by side. This one's only one strand, DNA is double strand, like a lot of that. And then it goes DNA, RNA, then from RNA to the actual protein. So this is really the main like overall steps of how the DNA will eventually go to protein. First, starting off with transcription. Here we go. So we remember back from another seminar that the DNA has to stay inside the nucleus. So what does that mean? That means that since the DNA cannot leave the nucleus, something else has to go inside the nucleus, copy what it needs to copy, and then it'll take that copy and then leave outside. If you think about it, let's say you have this super secret, top classified spy vault, right? And you can't take the vault outside the building, but you have some documents inside and you can open the vault, take the documents out, copy it, and then you can go away and then take it with you. So it's just like that. You can't take the physical thing and go away and go outside of the house, or in this case, the nucleus, but you can make a copy of it and then use that and then build proteins. And here we have the transcription is really the first part. The transcription is going to end up happening in this nucleus here, which is why I have the nucleus in the background. And the translation is going to happen outside the nucleus. Once again, this is going to be the transcription. And here, so I'm going to explain a little bit. The DNA is just going to unravel just a little bit, so only one section really. And then we're going to have this RNA that's going to be built, and it's going to be using like the matching nucleotides, right? So the only difference though is if you notice, you see these boxes right here, you're like, wait, why does A go with U? Remember in my last one, I said there's the one base that was changed. So in DNA, A goes with T, G goes with C, and that's adenine with thymine, guanine with cytosine. However, in RNA is different. Let's say that if the DNA has a T, it will still go to an A, right? But if the DNA has an A, it'll go to a U in the RNA. So we take a look right here. Okay, it's kind of hard for me to see, but we see this U right here, and then it's going to be bound to this A at the bottom. But we look over here, there's an A. Oh, excuse me, there's an A right here, but it's actually bound to a T. So just remember that when the RNA is copying it, any A's will become U's, and that's the only change. Here we have the RNA polymerase, and that's a special machine really that's going to be copying this RNA. And you'll see that this says like mRNA. Don't worry about it for right now. We're going to talk about it a little bit in the future, but just know that there's different types of RNA because of how they're used. So it's a little bit different. And this gives us our mRNA strand. Perfect. This can be like an analogy to, you know how you get in trouble? The teacher's like, I will not talk in class. Write it like 100 times. It's kind of like that, right? Because you're making a copy of it. Let's say the teacher wrote, I will not talk in class on the whiteboard. You're not going to somehow cut that out and then put that on your paper. No, you're going to use your hand and copy it, right? Just like with the transcription. With transcription, you're not taking it out of the nucleus, you're just copying it. And here's my next question is, how do we process the mRNA? Remember, it's just, you don't have to worry about it for right now. mRNA before it leaves the nucleus, right? So we have it, but it's still in the nucleus. And I'm hinting that we need to process it somehow. So we'll see. A, we have to cut out the unimportant mRNA. B, we add a tail of repeated A. C, we add a cap of guanine. Or D, all of the above. This one I kind of threw a curveball, added my first all of the above question. Give people a couple more seconds here. Okay, I think we can close the poll now. Ooh. Yeah, these questions are a little tougher now. So the correct answer is, drumroll please, D, all of the above. So we cut out the unimportant RNA, we add a tail of repeated A, and we add a cap of guanine. So I'm going to explain that too. This is our mRNA copy that we made, right? So this is before we really do anything to it. It's still kind of in the nucleus. We need to process it, right? Let's think about you're at some factory, right? And they're processing meat. Well, let's say you're having beef or something. You can't just take the cow and then just put it on a plate and just be like, take the live cow and be like, okay, now you can eat it, customer. No, you have to process it, you have to clean it, you have to put it in like a nice little box or like a bag like that so people can buy it and then go home. They have to cook it. So there's a lot of different steps, and that's kind of like this as well. So here we have these things called exons, and we have these things, oh, excuse me, sorry. The exons are right here, and we have the introns right here. So these are just two different ways to like classify the parts of mRNA. And what my little trick for memorizing it is, just remember exons, the EXONS, exons get expressed. And what does that mean? Well, we cut out the introns. That means we take them out. So that is going to be the unimportant mRNA that we were talking about. We cut them out. We're not going to use them to make our protein. But the exons get expressed, so we keep them. And many of you may be asking, why would you have the guanine like cap here and these like repeated A's, right? Well, it's actually very interesting because inside the cell, there's a lot of these just working enzymes that are doing a lot of processes in the cytoplasm, right? So if you guys don't know what that is, you can check out my first video on like cells, right? But we have all of these like enzymes working in the cell, and it's kind of like an open workspace. So we want the mRNA to go directly to like the rRNA, and like I'm going to talk about what that is, right? But we want to go there, right? And we don't need it to attach to some random enzyme, or we don't need it to get broken down. We don't want to break down our poor copy, right? We want to keep it. So what we do is very interesting, is they add a bunch of adenines at the end, which is the A's, to protect it, and a guanine at the top to protect it. These are going to act like, you could think about it like, let's say it's like freezing outside. You're not going to go outside, otherwise you're going to get like freezing, like, you know. So you need to put on like maybe a hat, and then also like boots, and then like a coat, just like that, because it'll protect you. Just like this, we need the guanine cap. So you can always think about cap as like the top, right? So the cap is going to be the front part. And then the tail is always the end. So it'll be adenine tail at the right side of the diagram. Here, same thing. Just see a little bit better. The color is a little clearer here. And we cut this out, put it in, they kind of put it together, and then we have the poly A tail. Poly just means many, so that just means many A tail. Here, we're going to get into something just a little bit complicated, but it's actually very interesting, because I'm pretty sure many of you are like, well, how do they just cut it out? You know, because we always know in biology, nothing just magically happens. That's why the answer wasn't like a magical wizard, right? Nothing magically happens. There has to be some process to actually do it and to actually cause it to happen. Well, great question, guys. Here, we have snRNPs, which I'm going to explain too, but you just need to know that what they do is they're going to go like underneath this intron, because they want to cut it out. They're going to like go underneath. So let's say this is the intron. This is the snRNPs. It goes underneath, it curls around, and it kind of like pushes and pulls it out. And then what happens is it brings, as it does that, it brings together the exons, and then they're kind of connected now. Then everything just disattaches. So let's say we start here, right? We push this out, the two exons come together, but then all of a sudden they just like, they fall apart here, which is okay, because then this part can go back. Same thing, push it out, exons come together, just like that. Here, we're going to explain what an snRNP is. This might sound really complicated. It's really not. So we look at small nuclear ribonucleoproteins. Very complicated, but it's actually not that bad. Because think about it, nucleus. They're in the nucleus, okay? Ribo, that means kind of like ribosomes, kind of like proteins as well. So we look back here, they're preparing it, right, for the RNA to go and go into like the ribosome. And yeah, they just they're like kind of like workers that are combining with this. They call it pre-mRNA because it hasn't been prepared yet. So it hasn't been prepared enough that it can actually leave the nucleus yet. That's why they call it pre-mRNA. So yeah, that's just something to know too. And that's really mainly it. And also, oh yeah, I want to point out one thing. This entire process here where you take, you take out the introns and you add the cap and the tail. This is called RNA splicing. And if you think about it, splicing makes sense. You're kind of like cutting something, maybe taking out some stuff and then putting it together. So it's a pretty good name. Now, this one's a little bit blurry, but I think it's okay here. Here we have the DNA, the RNA. So it just shows how the DNA went to RNA, right? But it just highlights the exons here, right? And the exons get expressed, remember that. And you may be wondering, like, what do they mean by alternative splicing? Well, we said RNA splicing, right, is where you cut it, you take out the introns, which are the stuff you don't need, right? The stuff that you're not actually going to need to express. You take that out, and then you put together the exons. Well, let's say for some reason, like, you don't want this one here, right, number 4. So you say, oh, I'm going to pretend that's like an intron. Then what can happen is you put all these together, you get a different set right here. You get a different set of the genes. And over here, so this one we're missing 4, right? Over here, we're missing 3. And over here, we have all of them. So I use the word missing, but it's not really missing them because in this case, for this protein, you don't actually need it. And by having different exons, which is different genes, right, that are copied to the RNA, by having those, we look at these proteins that are the end result, right? They're from the mRNA and they're produced. These proteins have different structures, right? So they're using this as kind of analogy. You see this one has a twist at the bottom. This one has a twist at the top. But then when we put both of them together, it has a twist at the bottom and at the top. So that's kind of how it works, and you kind of think about it like that. Do we have any questions? I know that was kind of a lot, so I want to make sure people have a chance to kind of take it in. And I think we can answer like one question.
Yes, we certainly do have a lot of questions. So our second question is, how can amino acids be negative? Does it have to do with electrons?
Yeah, so I may have to end up doing a seminar on amino acids as well, but if you look it up, and I guess for now you can look it up until I have a seminar ready, but you look at the structure, so the molecular formula, that just shows like the hydrogens, the carbons, and it kind of draws it. There's many different types of amino acids, and some of them are going to be like positive charge. Some of them are not really going to have charge, and some of them are like negative charge. And based off of that, that's how they can interact. Because think about it. We said opposites attract. If they were all the same charge, then nothing would happen. We would just still have the amino acids, right? And then the proteins can't do anything. Because we look that when it becomes more and more complex, and therefore it becomes like stronger and it can do more work, right? Just like this is called something called emergent properties. At each level, you gain some new properties, right? So we think about us. We're made out of cells. But one cell on its own can't do anything. It can do some stuff, right? It can like maybe take in a little food, like excrete some waste and stuff like that. But it can't be teaching you the seminar like I am right now. It can't be doing like water polo, playing piano. It can't do that because it's just one cell. But then you have a bunch of cells together working together. Then you have this organism that can do a lot of things and have all these functions, which is really an underlying idea in biology. That was an amazing question. I'm really glad you guys are all thinking about that too. Okay, we're gonna move on because I wanna really make sure I have time to really thoroughly explain everything to everyone, because I know this one is just a little bit harder. So yeah. Aha, finally. we get to the 3 types of RNA. This is messenger RNA. So this is the one, we go back here a little bit. Yeah, this is the one right here. mRNA has the RNA sequence, right? It's the copy. So mRNA, think messenger. What does a messenger do? It takes like a letter or something like that, right, to someone else. You can think about it, the RNA, mRNA, is taking a copy from one place and is going to another. Here we have ribosomal RNA. The ribosomes make proteins. Ribosomal RNA is what is here, right? We have the 2 main parts, and it's abbreviated as like rRNA. And so just remember, this is the actual ribosome. And over here, this is our amino acid, and this is actually connected to, you can see the 3 right here, that's a codon, since there's 3 nucleotides. So it's very interesting. It's almost like a key with like something at the end, right? Because you put it in and then you can have like the amino acid at the end. And that's called transfer RNA. You can remember this because you're transferring, right, from the codon, which are nucleotide, to amino acid. And it's kind of like locked together. So once you put it in, it's transferring it into an amino acid. Here are the RNA bases. So for the person that asked about like our amino acids, this is not really amino acid, but this is kind of the structure. As you can see here, you can see that like this is a nitrogen, and like there's a hydrogen up here, and there's like oxygen. And if you look at these rings, something to note too is that they're at each point here where it's not labeled otherwise. So unless it says like nitrogen or something else, this is a carbon. And so there's like a carbon like at each spot there. So it's pretty interesting. But again, just remember U goes with A, but G and C is the same in RNA. And finally, we go to the second step, translation. Okay, here is where we take the mRNA from our last step, put it in, take our ribosome, put it on top, and we begin to read it. Just like that, and polypeptide, don't worry, fancy word for protein. And this is actually making the amino acid sequence, right? Each one is an amino acid. We can think about translation as literally like translation, like in real life almost. Because you're going from, let's say, Chinese, right, to English, and you need to translate it. This is the same thing here. The mRNA is technically still like the DNA, RNA language, right? Because we still have these nucleotide bases. That's not what we want. We want the protein language, the amino acid language. That's what we want. So we got to go from this DNA/RNA into amino acid language to make the protein. So that's really where the translation comes in play. Okay, here we're going to actually talk about how this happens, right? So if you look here, we have, this is a tRNA, remember, transfer RNA, and it has the amino acid up here. We're taking out many different parts here, but we have the rRNA that has the 2 parts. So it has the bottom part and the top part that goes like this. The tRNA will go into this slot here, and then like it'll scoot over, but it's gonna match up right here with the codons, right? And so it has the corresponding, like as you can see, this one has GGC, this one has CCG, matches perfectly. Then what happens is, remember how like the mutations happen, right? Because it's going super fast. Well, you can think about it like the rRNA, so the ribosome, is just staying there. So it's just in one spot. However, the mRNA is just going like zoom, like really fast, like it's going super fast through it. And then the polypeptide is coming out the top, or the protein, excuse me. The protein is coming out the top. So just reading it super fast. Here, we can see one interesting thing is that each of the tRNA has 1 amino acid. So for each one, it has 1 amino acid. Here, it'll bind to this, remember, CCG with GGC. And then what happens is let's imagine that this is now right here in this spot. This PHE amino acid is going to form what's called a peptide bond, which is just a bond between amino acids. That's how they're all connected. And because if you think about it, right, technically it's called a polypeptide. And poly means many, and we're using peptide bonds. So it's pretty interesting. A lot of the names actually make sense if you think about it. And so that will create a bond like right here, and then that will add it to the chain. And then you just repeat the process. So after this bond is created, this one's going to leave. Here we have the 3 spots, EPA. So it first goes into the A slot, matches up. Then this amino acid here is going to form that peptide bond with this tryptophan, I believe. And this is going to create that peptide bond, kind of create the linkage here. Then this one, this tRNA, is actually not going to have an amino acid. It's going to like kind of drop it off because now it's already attached to this one. So originally we came in, this is the methionine one. It starts here, then we have the tryptophan. It's just a name for amino acid, so don't worry. Just the first amino acid, second amino acid, create a bond. Now the tRNA is going to exit. And then we scoot over, same thing happens, another one comes in, creates the bond here, then the other one scoots over. And it just keeps going, cycling. And so then the E is kind of like the exit spot, and then the P is where the active one is going to be, and then passing it on and then leaving. Then everything moves over. So I can definitely explain it at the end too if people are confused. This is a very cool chart, in my opinion. It might look a little complicated, but it's really not here. This is just called a codon chart. Here you see the word first letter, second letter, third letter. Let's go back to the example here, right? CCG. Okay, well, let's just use that, right? So we go up here, first letter C, second letter C, third letter is down here, G. So we look at the bottom. Oh, CCG. And then it says it goes to this amino acid. Now, what you might notice is, you might notice a couple things. First of all, why does it say stop? Is that an amino acid? No, it's not an amino acid. That is the one that I'm going to actually talk about a little bit that will actually stop it. Because you might think, okay, well, it gets started, right? But how does it eventually stop? Like, because it doesn't look like there's any way it's going to stop. Well, it'll actually get started with this first one called methionine. I'm just saying that because that's the very common one you'll run into. That's the very first amino acid, methionine. Then we have 3 stop ones, okay? And if you kind of think about it, it could be because like we really want it to stop. We don't want it to keep going. So that's why there might be 3, and those could be used all at one time to really make sure it stops. And yeah, so that let me actually go back one more time here. So let's pretend that, okay, in order to help everyone understand, I'm going to use the whiteboard for this, because this way it should be a lot easier. So we have the mRNA, then we have like our big ribosome RNA here. I'm just going to use this for like the 1 slot, the 2 slot. So the E, the P, and the A. Well, we already know how normally it works, right? But this we're going to pretend is a stop. This is going to be a stop tRNA. And it's going to be yellow to show it's different, right? And then we will use, let's just use nice red over here. This is just a normal tRNA. It's already dropped off, right? And so what's going to happen is this one is going out, and we have one. Okay, let me erase this really quick. So in the very center, remember, P slot, we have 1 tRNA like this, and then we have here. Let me try to move this a little bit. Okay, we have this is like these peptide, which is the protein, and it's kind of building up like that. Oh, I think I'm out of room. Okay, that's fine. And it has like all these little bonds here, right? It's like that, but we're done, right? That's all we needed to read. We're good. So what's going to happen is we're going to have this stop amino acid, and like it's not really amino acid, it's just a stop one. And then it's going to come in right here, and it's basically going to cause everything to actually, I'm not going to tell you what it does because I just remembered that's a question that I'm going to ask you guys, but it causes the process to stop in some way. And that's all you really need to know. And that's why that it's like very important for the process to stop, because we don't want to use extra resources to keep adding ones we don't need for a certain like gene. Okay, so I hope that really helped explain for everyone. Oh, I need to reshare my screen. Okay, here we go. And yeah, so again, you'll always start here, first letter, second letter, then third letter. So another example, let's say we have CUG. C, just go up here. U, that's fine. Second letter, third letter. So C and U, it'll be in this box. Then we look over here, G is the last row. Look in, CUG. And a key part of this is we see it's repetitive, but not redundant. What does that mean? Those are some very nice words, but I don't know what they mean, right? Well, what they actually mean is that you see all four of these, actually all six of these, all code for the same amino acid. So you're like, wait a minute, like, is that a problem? No, that's okay. It's just repetitive, right? But it's not redundant. Redundant means there's no reason to have so many, because if you look, no same codon codes for two amino acids. So let's say we take CCU, we look. There's no other CCU. And CCU will always go to this amino acid. It's never gonna go up here, it's never gonna go over there, it's never gonna go like this. So they're always fixed to that one amino acid. Many of them may go to the same one, but there will never be one that for some reason can have two. We go back, right? Each one has one codon, right? The three letters has one amino acid. And so it's never going to have two. That's just what it means. And again, keep that in the back of your head. It's just something interesting to really realize and know. Okay, now this is the question. So what does a stop codon do? A, nothing. B, it stops all the translation. C, breaks the system by ripping the protein. Or D, makes everything tangle in a mess. Now, you might be like, oh, I think it's just like stops, right? Because that's what he said. But like the question is, how does it stop? Does it stop it by breaking it? Or does it stop it by making it tangle? So that's just something to keep in mind as you put in your answers as well. Yeah, I always feel like this topic is very interesting because it's always cool to learn about DNA, but like how it actually relates to us, I feel is always cool because it shows directly how the DNA becomes like my hair or my hand, like that. Okay, I think we can give everyone like a couple more seconds and then we'll close it. Okay, we're gonna close it. So, wow, this one was also very tied. And again, I'm gonna reveal the results here. Is actually just stops all the translation. And you may be like, wait, Ryan, but what if it stops it by breaking the system? No, it doesn't break the system, ripping the protein. That'd be horrible because like, what would be the point of all the hard work? We did the splicing, we cut it, we made sure it's right? We made sure that like it's all ready, it doesn't get eaten by like the enzymes and the cytoplasm. We add the cap, we add the tail, we're like, okay, we're ready. And then we go and then we make it. And then our hard work, the protein, and we just break everything. And then so that'd be a very sad day, right? Or another thing is if it makes everything tangle, that'd be a huge mess. And again, it's almost the same thing as breaking it, because we're like, well, where's our protein that we made? And so now I guess I'll explain it a little bit more. So, wait a second. Let me start a new share here, and I'll draw the, oh, it's right here. So, okay, it'll go in, right? This is the stop codon. Yeah, it has a stop codon. It will go in, and it will just cause everything to calmly like separate. It's not gonna rip. It's not gonna tear. What will happen is we have, well, eventually this stop codon will actually also just separate, but we have like this big thing here. It's now in like 2 pieces, and then we have the chain here of the like polypeptide. Man, these look like blueberries, but yeah, the polypeptide, which is the protein. Remember, we just have that. That's all good. We have it. It's intact. It's not broken. We're good. And we also have just the tRNA, like normal. It's just over here. So we're good, successful. And so let's go back here. Okay. But let's take a look here at how this happens too, because if you think about it, couldn't we combine these, right? Well, let's take a look. So I'm going to put a note up here. This only happens in prokaryotes, and I'm going to explain what prokaryotes are and what eukaryotes are. Here we have the RNA polymerase, and it's reading the DNA at the same time. It's so efficient. At the same time it's reading it, it's making the RNA. But at the same time that it's making the RNA, it's reading it, it's making the protein as well. I don't know why this one says DNA. That's not correct. That's very not correct. I'm pretty sure you guys all know these are like the amino acids. So this one's kind of weird. But here we have the mRNA coming out, and then, oh my goodness, it is so efficient. Here we have this ribosome immediately start making protein. And remember that the ribosomes are staying in one spot. They're not moving up and down. So the only thing that's moving is the mRNA, and it's moving like through. So that's why you look over here, and this is the same part, right? But it just gets longer and longer, so more and more can fit. And they're saying like direction of translation, like what does that mean? But you look up here, these ones have the longest proteins, right? These ones have the longest proteins because it makes sense. They've been reading it for the longest. The ones at the bottom only recently attached. And the same thing happens. We have the stop codon, right? These protein, the made protein is gonna just remove itself. The ribosome is gonna remove itself from the mRNA, and the mRNA is gonna be done, so it's gonna remove itself from the RNA polymerase. However, what are prokaryotes, right? I talked about it a little bit, I think, and so I wanna ask you guys too, right? They might be a fancy name for red blood cells. Maybe that's why I chose this background. Maybe they're a vegetable. Maybe they're just a circular disc of animal cells, or they're just all cells without a nucleus. Yeah, and really like a prokaryote, it's very, very important because this is how we start to divide up like life, you know, as we know it. And we're kind of giving it in different sections and stuff like that. So it's very interesting. Make sure you guys cast your votes now, because we still have a good amount of the lecture to get through, and I wouldn't want to be waiting too long. So I think we're going to stop the voting. Okay, you guys are picking up speed again. All cells without a nucleus. Good job. Great. Here, prokaryote, single-celled organism, no distinct nucleus, membrane, or specialized organelles. Yeah, that's good, but like here's what the main thing is. Prokaryotes, they don't have like the nucleus, remember? So you know how the nucleus is like this big kind of ball thing, right, that has the DNA? They just don't have that. They still have DNA, they still have like genetic information, they still have it. They just don't have that big, like the ball housing it, right, really. And so you may be asking, like, does it just float around? Well, we're gonna find out in like the slide right after this where I compare the two. But moving on to the next question, what are eukaryotes, right? We know prokaryotes now, so hopefully you have a better idea of what a eukaryote is. A fancy name for the color red might be a type of fruit this time, not a vegetable. Or it could be a frisbee. Let's see. Yeah, we're not going to spend that much time anymore on these like polls, just because we really need to like keep going through the lecture. Okay, and again, you guys are doing great. All cells with a nucleus. And how I really remember this is you think about our cells, right? Animal cells, we have a nucleus. We're eukaryotes. Think about the name eukaryotes. Eukaryotes. So that's how I remember it. I'm like, oh, it's me. I have a nucleus, right? So it's eukaryotes. Great job. Here, organism consisting of cells in which genetic material, DNA, and it has chromosomes and a nucleus. Okay, finally, we have some image. So you hopefully already know what a eukaryotic cell is. I talked about that in my cell seminar, so hopefully you're paying attention to that. And we can see nucleus right here. However, we look at this, and this thing's like a very interesting shape, okay? It has a cell wall and like a capsule that's like surrounding it. So it's a very different structure. And it also has this thing called the flagellum. It's just a tail. It's just a tail. That's all it is. Ribosomes, cytoplasm. Okay, we know what those are. The membrane. Okay, we've seen that from the other ones. And the pilus is these just little like these little arms kind of coming off as well. Here, it's just like DNA. It literally looks like a ball of spaghetti just in the middle, just like floating. So you can see that there's really, it doesn't seem to be that much like order and organized as it is here. And also, what about the mitochondria? What about like all these other complex organelles that we have and they don't? And this is why many like prokaryotic cells usually just like one cell. They can't really get bigger because they don't have the machinery, they don't have the organelles to really do much. Because we're glad we can get really big and get really large and everything because we have proper systems from the foundation. We can break down stuff. We can make proteins. We can do a lot of stuff with like our cells, but their cells can really do just the bare minimum. Okay, just one more overview. This is inside the nucleus. Transcription, always in the nucleus, remember that. Then we copy it because we can't take out the original. Have the mRNA actually right here. We splice it, and then it leaves, right? We have the guanine cap, poly-A tail, which just means many A's. Then over here, mRNA. This is another way to show the tRNA, transfer RNA, remember? Transfer RNA has the amino acid connected to the 3 letters here, codon. And here is the growing amino acid chain. That just means you're moving it over and it's growing larger. And then another one comes in, you're moving it over, growing larger. tRNA is going out this way. And again, the ribosome is not moving. It's staying still. And it's like this is a really good analogy, subway station. If you're in the underground subway, you're standing there waiting for the train, you're not moving to catch up with the train, or the train is just like stopping. The train is moving. So that's just like the mRNA. You're staying here, the mRNA is going through, just like that. Okay, now do we have, actually, I'm going to skip this question slide. I know we don't have that much time, and I still want to get through a lot of this really important stuff too. Cool, but like, why do we need to know this, right? So translation, transcription. Well, here's the main idea. Certain gene, remember, segment of DNA. So like DNA, but like how many long is a gene? Makes a certain protein, which makes how we look, right? Another example I used was like attached or free earlobes. Well, if we get it from our parents, that means it's in our genes. And then it will use this process to make the protein, and then the protein will actually make this shape, which is how we look. So I think that's why it's so important. Here, The same sort of overview here. I'll just let you guys kind of look at that for just a little bit here. And what's a very interesting thing is on the right, though, we see a little bit of a kind of different thing here. A very interesting to know is that inside the nucleus, the DNA is not always like that DNA structure. Sometimes it's this thing called chromatin, and it's very like stringy as well. And so it'll change. It'll like condense, become the DNA. It'll sometimes like kind of uncoil a little bit and be kind of messy. But really, when it needs to be like chromosomes, right? Let's say for the division, it'll condense a bunch and become chromosomes. But let's say that it doesn't need to divide. How is it going to use the genes, right? We look here, it needs this DNA segment. It can't be super coiled. There's no way, right? Because how else is the mRNA going to be made if the DNA is super coiled and you can't read it? You can't see, oh, wait, which nucleotide is here, right? So it needs to uncoil really for a lot of the processes that happen. And here we're going to actually talk about, here's like a little bit of the different kinds of mistakes that can happen. We're going to have the substitution. So you look that, oh, this was the wrong one, right? It should have been a G, but the machine actually accidentally put an A. And then what's going to happen is, oh, excuse me, sorry. In the DNA, it was actually supposed to be a C. Then it is now T that causes the RNA to be an A. And then that'll change the amino acid. Well, remember, sickle cell anemia, it was one change. And it changed the amino acid. So that's a pretty big deal, right? But then what's even a bigger deal is you have an insertion, right? So let's say, like, I don't know, you have the sentence, I like spending time with my friends. But then you move all the letters over one. All of a sudden, you can't read anything. But let's say for the substitution, you substitute one letter out, right? I lime spending time with my friends instead of like. Well, you can still understand, and there's still, you're like, oh, something, friends. But you move every letter over one, you can't read it. It's impossible because it can be like blah, 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 blah. Like you can't read it. That's why this is such a like really bad mistake to happen, because it'll change the amino acids, and then like really like all the amino acids in the future from this point on, because after the space has been added, everything is shifted over. All the amino acids are going to be wrong in the future from this point on this segment, right? Here we have a deletion, same thing, except this time it kind of goes the other way, and everything is kind of going to be wrong because the amino acids and the DNA was just really messed up like that. Here I have a video. I'm going to actually stop sharing, I think, because I need to choose the share computer sound with this. And this one is a really good video because I think it's great, just really showing what we talked about. And I'm going to do it at a little bit of a higher speed, so that way, because again, we still need to get through the questions and everything.
The events going on in our cells can be compared to an orchestra playing a symphony. There are many different parts being played by a wide range of instruments. Here we see an example of a protein in short supply found in the external environment of the cell. Proteins interact with specific receptors on the cell's outer membrane to trigger a cascade of events leading to the expression of specific genes and the production of this protein. Membranes regulate the movement of substances in and out of the cell through a wide range of mechanisms. In this case, receptors on the surface trigger signaling mechanisms involving other proteins in the cell's cytoplasm. In the cytoplasm, the signaling elements are involved in a cascade of events. The final components interact with the DNA in the nucleus at very specific sites to begin the expression of the desired gene. Gene expression is complex. To start transcription, several proteins are often required to bind regulatory elements on the DNA. This step in the process is where DNA is read and a complementary strand of RNA is produced. The RNA has to undergo many changes, including the systematic removal of RNA sections not needed to code for the desired proteins. RNA then takes the information encoded in the DNA out of the nucleus. In a process called translation, ribosomes interact with the messenger RNA and transfer RNAs to create the chain of amino acids coded for on the DNA. This amino acid chain undergoes changes in configuration, including specific folding. Each protein takes on a 3D shape required for a functional end product. As the new protein leaves the cell and takes on its role in the body, the cycles of cell function and gene expression carry on.
Yeah, so I think that was like a really good one to really summarize everything we learned about. So yeah, I'm really just like excited, and I thought that was a really good wrap-up video. I have a pretty big note here. Next class is actually going to be on Friday, August 7th from 6 to 7 p.m. if you're in Pacific Daylight Time, or Saturday, August 8th, 9 to 10 a.m. for Beijing time. So that's just a note for my next class.
Thank you all for coming today, and thank you for sticking with us to the very end. And again, big thank you to Ryan for this seminar. It was very informative and entertaining as well. All right, so thank you to all of you, and have a great day, you guys.
Thanks, everyone. Hope to see you guys soon.